Welcome back to the deep dive. Our mission is to take complex source material, distill the core wisdom, and turn it into knowledge you can use.
And today, we are definitely diving deep.
We're tackling the draft ICH Q3E guideline for extractables and leachables, E&L. For anyone preparing for, meetings or an exam, this is the guideline where analytical chemistry and toxicology are finally forced to shake hands.
That is the perfect way to put it. And that's really the key insight. This isn't just another document about impurities. Q3E is about formalizing a risk-based strategy. It borrows heavily from ICH Q9, from quality risk management principle.
All to make sure that the things used to make or package a drug don't hurt anyone.
At its core, yes, that they don't compromise patient safety.
Okay, so let's unpack this with the absolute fundamentals, because the terminology here is critical. You hear extractables and leachables.
All the time.
And they sometimes get used interchangeably. But in the lab, and certainly in the eyes of a regulator, they're distinctly different things.
Oh, completely different. And you have to get this right. We always start with the actual risk, and that is leachables. The L.
Okay.
Leachables are the chemical compounds that actually migrate from, say, the manufacturing equipment, or more likely the container closure system.
Like the vial or the stopper.
Exactly. And they migrate directly into the finished drug product. This happens under normal, real-world storage and use conditions. So leachables are the actual impurities the patient is exposed to.
So if leachables are the confirmed actual impurities, then what are extractables?
Extractables, the E, are the potential impurities. They're what you get when you take a component, that rubber stopper or a plastic syringe, and you hit it with a hammer, metaphorically speaking.
In the lab?
In the lab, yes. You use aggressive solvents, high temperatures, conditions far harsher than real life. And you do this to intentionally pull out everything that could possibly migrate out. It's your worst-case chemical profile.
That distinction is so important. So extractables define the potential hazard, and leachables confirm the actual risk. Now, where does this Q3E guideline fit into the bigger ICH picture?
It slots right into that core Q3 framework for impurities. I mean, if you've worked in this space, you know Q3A and B cover impurities and the drug substance and product.
Right, and Q3C for solvents, Q3D for metals.
Exactly, and M7 for mutagenic impurities. So you can think of Q3E as the dedicated chapter for organic impurities that come from these materials, the plasticizers, the antioxidants, things like that.
So it's the official organic E&L chapter.
It is, but it's important to note, while Q3E gives us the methodology, it specifically defers the safety assessment for any elemental leachables, metals, back to Q3D.
And its scope is pretty wide, right? It's not just for new drugs.
Oh, very wide. New drug products, cell and gene therapies, drug-device combinations, and critically, it also applies when you make a change to an approved product. It forces you to think about your materials from day one.
Okay, so this is where it gets really practical. The entire Q3E structure seems to rest on two foundational concepts: the safety concern threshold, or SCT, and the analytical evaluation threshold, the AET.
They are the two pillars of the whole guideline.
So let's start with the safety bar, the SCT. What is it?
The safety concern threshold is, it's the toxicological linchpin. It is the exposure level below which a leachable is considered to present a negligible safety concern.
Negligible. So that means we've accounted for all kinds of toxicity.
All kinds, both mutagenic, which is, DNA damage, cancer risk, and non-mutagenic toxicity. It's the highest daily dose of an unknown compound we're comfortable saying is acceptably safe.
Okay, but how do you arrive at that number?
This is a critical point for anyone learning this. The SCT is always determined by the most protective, so the lowest, value from two different kinds of thresholds.
Walk us through those two.
Okay, so first we have the threshold of toxicological concern, the TTC. This is borrowed from ICH M7, and it is all about controlling mutagenicity, cancer risk. And second, you have the qualification threshold, or QT. This one is focused on non-mutagenic toxicity. It's the dose where we think systemic effects are negligible.
So you calculate both, and the SCT is just whichever number is smaller, the more conservative one.
Precisely. The guideline mandates that protective approach. We can make this really tangible if we look at the chronic parenteral exposure data. So for treatments lasting longer than 10 years.
That would be the highest risk. Longest exposure, so you need the tightest control.
Right. So for that route, the qualification threshold, the QT for non-mutagenic effects, is 12 micrograms per day.
Okay, 12 micrograms.
But the threshold of toxicological concern, the TTC for mutagenicity, is much, much lower. It's 1.5 micrograms per day.
Wow. Okay, so in that case, the SCT is automatically 1.5 micrograms per day. The 12 micrograms is just it's too high when you factor in the mutagenic risk.
Exactly. That tiny 1.5 microgram limit becomes the safe daily dose, and that number dictates everything that happens downstream, especially how sensitive your analytical methods need to be.
So we've set the safety bar with the SET, but how do we translate a theoretical safe dose into something our lab instruments can actually measure? And that brings us to the AET.
The analytical evaluation threshold. Yes, this is the analytical benchmark. It's the concentration threshold where if you find an extractable or a leachable above it,
You have to investigate.
you have to investigate.
Yeah.
You must identify it, quantify it, and report it for safety assessment against the SCT.
So the toxicologist sets the SCT, the safety bar, and then the analytical chemist uses that to calculate the AET to make sure their lab methods are sensitive enough to find anything potentially dangerous.
That's the relationship in a nutshell. And this is why we have to stress, and you should underline this for any exam, the AET is not a control limit.
Right. Seeing something above the AET doesn't mean your batch fails.
No.
No.
It just means you now have to do the work to identify it and prove that its exposure level is safe when you compare it back to the SCT.
I think we should slow down and really nail the calculation part here. When we talk about AET units, what are we even measuring?
Well, it can be a few things. You might express it in micrograms per gram of a component, or maybe micrograms per milliliter of your drug solution. And you'll often hear it talked about in relative terms, like parts per million.
And we should say that out loud? Parts per million, not ppm.
Correct. Parts per million. It's just a common way in chemistry to talk about a very small concentration.
Okay, let's use the guidelines example to make the math real. Say we have a pre-filled syringe, and we need the AET in micrograms per milliliter of the drug product.
Okay. So the basic formula is this. AET equals the SCT multiplied by an analytical uncertainty factor, and then you divide that whole thing by the maximum daily dose in milliliters.
All right, hold on. Why do we need that analytical uncertainty factor? Let's call it the UF. Why not just use the SCT directly?
That is a fantastic question. And it really gets to the reality of ENL testing. A lot of the analysis we do is semi-quantitative.
Meaning you don't have a perfect reference standard for every unknown peak you see on the chromatogram.
Exactly. You often have to use a surrogate standard or a general response factor, and that introduces uncertainty. Your instrument might be less sensitive to that unknown compound than you think it is.
I see. So the UF is a safety buffer.
It's a mandated, built-in safety buffer. It's typically 0.5 or less. And what it does is it deliberately lowers the AET. It makes your analytical target more challenging to account for that potential underestimation. You're basically saying, we have to be able to find this thing even if it's at half the calculated safe level, just in case our measurement is a bit off.
And the guideline is also very clear that you have to use the maximum daily dose and the minimum potential batch size. You always plan for the worst-case scenario.
Always. You have to protect the patient who takes the maximum allowed dose. And if you're calculating the AET based on a component, you have to assume the smallest possible batch size, because that means any leachable is concentrated in the smallest volume of drug. You always default to the most conservative case.
That makes sense. Which brings us to practical application. We have the data, we have the thresholds. How do we apply it all in a risk assessment?
The strategy is really based on a multidimensional risk matrix. You look at pharmaceutical quality factors, for instance, liquid dosage forms are almost always higher risk than solids.
Yes.
And then you look at safety risk factors, where the route of administration is probably the single most critical driver of risk.
And the guideline specifically flags some routes as being particularly high risk.
It does. Injectables, inhalation products, and anything that directly contacts what it calls vulnerable tissues. So we're talking about intracerebral, intrathecal, epidural, and ophthalmic injections.
So if the standard SCT is designed for systemic safety, what happens when you're injecting something directly into the brain or the eye?
Well, that's where you hit a much higher regulatory hurdle. Because tissues like the central nervous system have very limited capacity for repair, the guideline says that for these routes, the generic SCT is often not enough. You might be forced to justify limits down in a very low parts per billion range.
Parts per billion. That must just explode the complexity and the cost of an analytical study.
It absolutely does. When you're talking parts per billion, you've essentially eliminated the use of generic thresholds. You're forced into doing expensive, compound-specific toxicological work, even for tiny amounts, because that local toxicity risk is just too high.
Let's talk about the leachable classifications. The guideline sorts them into 3 categories, which helps organize the risk.
It does. For toxicologists, this is key. First, you have Class 1 leachables.
The known bad actors.
The known bad actors, exactly. The compounds you must avoid. They have highly potent toxicity, either mutagenic or non-mutagenic. A classic example would be benzoaperine, which can come from carbon black, or maybe bisphenol A, BPA from certain plastics.
And if you can't avoid them.
You have to set a much lower compound-specific safety limit. You can't use the generic SCT.
Okay, so that's Class 1. What about the low-risk compounds?
That's Class 3. These are compounds with relatively low systemic toxicity. We're talking about things where the permitted daily exposure is 1 milligram per day or even higher.
So things like
Common antioxidants like BHT or long-chain fatty acids like palmitic or stearic acid. For these, if the patient's exposure is under 1 milligram per day, they're generally considered qualified without more work.
And Class 2 is just everything else.
Correct. Class 2 is the default. If it's not a potent Class 1 compound and it's not a low-potency Class 3, it falls into Class 2. And you control it using that standard SCT we calculated earlier.
Let's shift to data. You've done your extractable study, you've done your leachable study. What's the goal when you try to correlate the two?
The correlation is, in many ways, the ultimate justification for your control strategy. The goal is to prove that what you see in your leachable study is a subset of your extractables.
At a much lower level.
And at much lower levels. If you can show that strong correlation, it can save you a massive amount of resources down the line. Well, it can support using a routine extractables test as a quality control check for your incoming components. So instead of doing a long, expensive leachable study on every single stability time point, you might be able to qualify a new batch of stoppers, say, with just a quick extractables test, because you've already proven that relationship holds.
This brings up a common pitfall, right? The difference between manufacturing components and packaging components.
A huge one. The risk for manufacturing components like, say, tubing or filters is generally lower. The contact time is short, and there are often purification steps afterwards that would remove any leachables.
So you might get away with just extractables data for those.
Often, yes. You can frequently justify qualification based on extractables data alone, as long as all your peaks are below the AET and you don't see any Class 1 compounds. But the primary packaging, the container closure system.
That's a different story.
A completely different story. It's in contact with the drug for its entire shelf life. It almost always requires full, dedicated leachable studies on the actual drug product over time at different stability conditions. You need that real-world data.
One last point on this. What happens when you find an unknown? A Class 2 leachable, maybe?
Yeah.
But there's just not enough toxicology data out there for it.
The guideline encourages what it calls a read-across approach. So if data on your specific compound is sparse, you try to find a structurally similar compound, a surrogate, that does have good toxicological data. And if that surrogate's data supports the safety of your leachable at the level you found it, you may be able to avoid doing new, expensive safety studies.
That feels like a very practical acknowledgement of the reality of this work.
It is. It's about leveraging existing knowledge to be more efficient.
Okay, that brings us to our essential takeaways. If you remember nothing else from this deep dive for your next exam or that big regulatory filing, these 5 points are what you need.
Point 1, leachable versus extractable. L are what actually migrate into the drug. They're the real risk. E are what could potentially migrate. They define the hazard.
Point 2, the SCT equation. The safety concern threshold is a minimum of 2 values.
Yeah.
The TTC for mutagenicity and the QT for non-mutagenic toxicity, always the lower of the two.
3, AET's role. The analytical evaluation threshold comes from the SCT. It sets the benchmark for what you need to identify and quantify. But, and this is key, it is a reporting limit, not a control or failure limit.
4-5. Risk drivers. Your risk is highest with chronic exposure, high daily doses, and those high vulnerability routes, injection, inhalation, and especially things like intracerebral delivery, which can push your limits down into the parts per billion range.
And finally, 5. Classification significance. You must avoid or have very tight specific controls for Class 1 leachables like benzoaperine. And Class 3 leachables like BHT or fatty acids are generally low risk if the daily exposure is below 1 milligram a day.
This guideline really does seem to force that cross-functional conversation between the analytical lab and the toxicology group.
It absolutely does. The science demands that collaboration. You can't do it in a silo.
So as you continue to explore this field, here's a final thought for you to mull over. The guideline introduces this idea of using a read-across approach for safety when specific data is missing. So, how does relying on a surrogate compound, no matter how similar it is, fundamentally change the margin of safety we rely on for patient protection? And how much uncertainty are we willing to accept in exchange for avoiding those extensive product-specific toxicology studies?
That trade-off right there, between speed and absolute certainty, that is the core debate that will continue to drive this guidance forward.
Thank you for joining us for this deep dive into ICH Q3E. We hope you feel much more prepared to navigate this crucial area of pharmaceutical science.